Hi, this video contains exam questions on the Amylase required practical. As you go through the video, pause the video, answer the questions, and then mark your answer based on the mark scheme provided.
Amylase can only digest starch. Explain why amylase cannot digest other substances. This question is worth 2 marks. Pause the video and have a go at the question. So the reason why amylase cannot digest other substances is because the enzyme has an active site which has a specific shape that will only bind to the substrate, which in this case is starch.
Students investigated the effect of pH on the rate of starch digestion. This is the method used. Add 2 cm³ of amylase solution at pH 5 to a test tube in a water bath at 37 degrees Celsius. Add 2 cm³ of starch suspension to the same test tube. Start the timer. Remove a drop of the amylase starch mixture after 30 seconds. Test the drop for starch. Repeat steps 4 to 5 until no starch is detected. Record the total time taken for no starch to be detected. Repeat steps 1 to 7 using amylase solution at different pHs. Which is a dependent variable in the investigation? Tick one box. Pause the video and have a go at the answer. The answer is the time taken for no starch to be detected.
Describe the test for starch. Give the result of the test if starch is present. You have to give the test and the result. This question is worth 2 marks. The test that you would use is iodine solution and it would turn blue-black. The amylase solution and starch suspension were kept in the water bath at 37 degrees Celsius for 10 minutes before being mixed. Why were the amylase and starch kept at 37 degrees Celsius for 10 minutes before being mixed? This was to ensure that the starch and amylase both reached the same temperature, which was 37 degrees Celsius.
The table below shows the results. Student A concluded that the optimum pH for amylase is 7.5. Give one reason why this conclusion could be correct. Use the table above. The answer is at pH 7.5, the least time was taken for no starch to be detected. Student B concluded that the optimum pH is between 7.0 and 8.0, but may not be 7.5. How can student B determine a more accurate value for the optimum pH? Use the table above or the table that was mentioned in the previous slide. The student should repeat the experiment using smaller pH intervals.